use food, and our microbiome, which is the good bacteria that live in our bodies, and even cannabinoids to modulate and treat IBD. In today's Science Cafe, our two speakers will talk about their work on IBD and specifically how to design diets and probiotics which can rebalance the gut microbiota in order to help treat this disease. And before we get started with introducing our speakers, I just wanted to quickly mention a couple of things. So the first one is that the views in this presentation and on the following slides are solely those of the presenter and not necessarily those of the UMass Chan Medical School. Also, as you may have noticed, this session is recorded and the video recording will be available later and will be posted on our YouTube channel. And we will share the link to this recording to everyone who RSVP'd. Also, if you have questions during the session and during the Q&A session, please type them in the chat box and we will ask them for you during the Q&A. And also we have social media channels. We have Facebook, YouTube, Twitter, Instagram, and LinkedIn. So please follow them and we will post the links to those in the chat shortly. So with that, I will now go ahead and get started with introducing our speakers. So our first speaker today is Dr. Mario Meza-Segura, and his LinkedIn is posted here. So if you would like to follow him on LinkedIn, please do so. Dr. Mario Meza-Segura is an instructor in Dr. Beth McCormick's lab here at UMass Chan Medical School. He got his PhD in molecular biomedicine in Mexico, where he had the opportunity to characterize different intestinal pathogens associated with severe cases of acute diarrhea in children. His work with infectious diarrhea captured his interest in the mechanisms regulating intestinal inflammation and motivated him to transition to study inflammatory bowel disease, or IBD. When outside of the lab, Mario enjoys spending time with friends, and when it's warm outside, going for long bike rides around New England. Mario, welcome to our Science Cafe series, and you have the mic.
Hey, hi, everyone. As Alicia mentioned, my name is Mario Meza, and let me share my presentation. Okay, so share it. Oh, I cannot share it. Can you allow me to do it?
You should be able to share it now.
Okay. Yeah, I think I can now. Okay, so as Alicia mentioned, my name is Mario Meza. I am a postdoctoral associate working for Beth McCormick in the Department of Microbiology and Physiological Systems. And today, I'll talk to you a little bit about the basics and not so basics of inflammatory bowel disease. So to start with this, I would like to start by talking about one of my favorite subjects, which is diarrhea. So diarrhea is defined as the passage of 3 or more liquid stools in a day or with a higher frequency than what is normal for an individual. Can you see it fine? Okay. To what is normal by the individual. And depending on the duration of the symptoms, diarrhea can be classified into acute, which lasts usually between a few hours to a couple of days and usually doesn't require any treatment. Well, this kind of diarrhea can be non-infectious and result simply by changes in our diet or the use of antibiotics. It can also result from an infection with different pathogens, including viruses, bacteria, or parasites. However, there is a second type of diarrhea known as chronic because it lasts longer than 4 weeks, and it is characterized by presenting episodes of symptoms that can come and go regularly. Within this group of diarrhea, we can find 2 very important conditions, which are irritable bowel syndrome or, wait, give me a second, irritable bowel syndrome or IBS, and inflammatory bowel disease or IBD. And while these 2 conditions share some similarities among them, such as the fact that both are chronic and they are characterized by the presence of abdominal pain or diarrhea, they are 2 very different conditions regarding their symptoms and the way in which they are treated. For example, IBS is not really considered as a disease, but instead it is a functional disorder, meaning that although everything looks good in the gastrointestinal tract, it is not working properly, and it results in symptoms, in the presence of symptoms such as cycles of diarrhea and constipation, the presence of gas, trouble sleeping, and bloating, which is defined as the presence of having an inflated balloon in our belly. However, it must be highlighted that despite these symptoms, it has been well characterized that IBS does not result in tissue damage in the large intestine. On the other hand, IBD is an inflammatory disease that results in destruction and permanent damage of the intestinal tissue. Patients suffering from IBD often present symptoms such as fever, the presence of blood in their stool, and weight loss. And it is important to highlight that in contrast to IBS, IBD can get progressively worse, and it can result in an increased risk of suffering, of going into surgery or getting colon cancer. Despite all these complications, it is, in addition to this, it's important to mention that IBD has become an expanded global health problem in the world because of its rapid increase in incidence. While the increasing incidence of IBD, the incidence of IBD increases slowly after the first case was reported in 1859, it exploded by the last 50 years of the 20th century. And it changed from only a few thousand cases affecting a couple of countries to more than 6.8 million cases reported in 2017, affecting mainly westernized countries. So this leaves clear the urgent need to better understand IBD incidence in order to prevent the increase, the rise in IBD. So to get it more local, now let me ask you a question. How many people do you think suffer from IBD in the United States? Please answer the polls that will appear on your screen, and we'll review the questions, the answers shortly.
We're going to launch the poll in just 1 second.
Yep, we have it going.
Okay.
Yeah, it's going. I can see the numbers.
Great. We're going to give it like maybe 10 minutes, 10 seconds more, and then
Yeah.
we'll see the results.
Okay, so most people voted for 3.1 million, and let me tell you that, yeah, you're right. So according to the CDC, in 2015, it was reported that more than 3.1 million people in the United States suffer from IBD, which represents 1.3% of the adult population in the country. So therefore, because of all this increased incidence in IBD and all the complications, many scientists around the world have been working on understanding the causes of IBD. However, despite their efforts, up until now, we have not been able to identify the exact causes of IBD. Nonetheless, thanks to these studies, we know now that IBD is a multifactorial disease with the three main factors being a genetic susceptibility, as it has been noted that patients suffering from between 5 to 20% of the patients suffering from IBD have a first-degree relative that has also been diagnosed with this disease. There are also some environmental factors that contribute to the development of this disease, such as air pollution, stress, and the lack of sleep. And finally, we need to be careful with the food that we eat because it can also influence in the onset of IBD. And it has a major impact on the health of the microbes that reside in our intestinal tract. But this will be explained later by Anna, so I won't mention much about this. But taking together all this information, in general, we can say that IBD is the result of an uncontrolled immune response in genetically susceptible individuals. But what do we mean when we say that this is an unregulated immune response? For that, first we need to understand how the immune system works. And as some of you know, in our day-to-day life, we are continuously getting exposed to different pathogens, which can eventually lead to an infection, making us sick. However, there is a specialized group of cells in our body that are known as white blood cells or leukocytes, and they are in charge of defending our body against these pathogens in a process known as inflammation. While inflammation can be is necessary to protect our bodies from intestinal pathogens, it is important to mention that when this process gets out of control, there is an increased recruitment of some leukocytes known as neutrophils to the intestinal lumen. And while these leukocytes are trying to get rid of the pathogens, they can also cause damages to the intestinal tissue. So while we still don't fully understand what triggers IBD, we know that an uncontrolled immune response, such as the one I just explained to you, is responsible for the onset of the disease. So now that we understand how IBD is developed, it will be easier for us to understand how the diagnosis of IBD. So for IBD to be diagnosed, first, we need to start by reviewing the medical history of the patients and to discard the presence of an intestinal infection being responsible for the symptoms. Once this has been done, the patient needs to go through a physical examination, and then blood samples and a stool are collected to analyze the presence of inflammatory markers. Based on these results, we can determine if there is the presence of inflammation, and once this has been established, your physicians may ask you to go through through other studies, such as endoscopies and the collection of biopsies. An endoscopy is a procedure in which we use a camera to analyze how your colon looks inside. And based on the results from an endoscopy, physicians can determine if there is the presence of inflammation, where it is located, and the severity of the disease. So thanks to endoscopy, we can know how the healthy colon looks like. And in here, I'm showing you a picture of a healthy colon. And by analyzing the samples, we can look at the normal architecture of the intestinal epithelium. However, during IBD, there is inflammation and ulceration of the tissue. And we can see a disruption of the epithelium characterized by a massive infiltration of neutrophils. And usually, the severity of IBD is characterized by how much of the colon is inflamed. So depending on the characteristics of IBD, we can subdivide this disease into two different conditions known as ulcerative colitis and Crohn's disease. They are very different conditions characterized particularly, for example, in the case of ulcerative colitis, because it only affects the large intestine, also known as the colon. It usually starts in the rectum and extends from there in forming a continuous area of inflammation. On the other hand, Crohn's disease can affect different regions of the gastrointestinal tract, going from the mouth to the anus, and it presents in patches of inflammation. It's important to highlight that ulcers observed in Crohn's disease can penetrate the entire thickness of the gut. So once IBD has been diagnosed, it is necessary to implement the right treatment in order to relieve the symptoms, to induce remission, which is known as the state of IBD, in which the disease is no longer active, and also to prevent relapse and to reduce the risk of complications. So treatments of IBD usually involve a combination of nutritional support together with drugs or surgery. It usually starts by using anti-inflammatory drugs that are used to treat mild colitis, but it can also include the use of short corticosteroid treatments in order to bring the patients into remission. We can also use different drugs known as immunomodulators can be used to reduce inflammation and to prevent the establishment and to prevent more inflammation. However, if none of these treatments work, not even in combination with a diet, the patient needs to go into surgery in order to alleviate the symptoms. And these processes usually involve the remotion of parts of the colon. In this case, I'm showing you a colostomy, which is in which a part of the colon is removed and connected to a bag where the feces are collected. So in order to prevent all the damages caused by IBD, in our laboratory, we are focused on using new molecules to modulate inflammatory responses. And based on the finding that neutrophils are very important for this disease, we are focused on using cannabinoids to modulate the migration of neutrophils to the colon. And some of you may have already noticed that, well, may have already identified the word cannabinoids, because and these are lipids that can be either produced by our body and are known as endocannabinoids, or it can be found in plants. For that reason, they are known as phytocannabinoids, which are and in this group we have the ones produced by plants, such as marijuana. So we are focused on using these different compounds, and we know that these cannabinoids can be sensed by different receptors, such as the receptor CB1, which is responsible for the psychotropic effects of the marijuana. However, there is a second receptor known as CB2 that is found also in neutrophils and is responsible for the modulatory effects of these endocannabinoids. So now we're interested in identifying those cannabinoids that can also be sensed by the CB2 receptor and use them to modulate the symptoms observed in IBD. And to do this, we have developed a model in which we can use freshly isolated human neutrophils to analyze their capacity to migrate due to an inflammatory signal that we add to the bottom of these chambers. And by using this model, we have been able to demonstrate that an endocannabinoid known as anandamide is able to completely inhibit neutrophil migration. But also we have identified other cannabinoids, such as CP55, which is a synthetic cannabinoid that is able to block neutrophil migration at a maximum of 50%. Similar to these two compounds, we have already analyzed a collection of other endocannabinoids, and now we are interested in analyzing and using in vivo models of colitis if we can combine these compounds in order to ameliorate the symptoms of IBD. So as conclusions for this talk, I just want to say that it's important to remember that IBD is a global health problem due to its high incidence and the complications associated to this disease. And if it's important that if you experience persistent diarrhea or other symptoms associated to IBD, that you go and visit your doctor. So in case you want to know more about this disease, you can visit this website that is very informative, and it's going to guide you through all the steps that you need to know during this disease. And I just want to mention a final disclaimer that the data that I show you today is part of an ongoing research, and the cannabinoids that we have tested are not part of a clinical trial or have not been approved by the FDA as a treatment for IBD. So finally, I would just want to conclude by thanking everybody that has been involved in this project. Please let me know if you have any questions.
Great. Thank you, Mario, for the fantastic talk. So we'll now start with the Q&A. And so if you have questions, please type them in the chat box, and maybe I'll get us started with a question. So you mentioned the massive increase in incidence of IBD. So I'm just curious if it's known why that has gone up. Is it antibiotics? Is it the hygiene hypothesis, or what's happening?
So basically, it is linked to industrialization. That's what has been described so far. That while more people are, industrialization is linked to more people migrating to the big cities. So while more people were migrating to the big cities, also feeding habits were changing. And that is basically what has been linked to the increase in incidence. But the real causes, we don't know. Like, there are many different factors that can influence in this.
That's interesting. Thank you, Mario. That was a very interesting talk. We do have a question from Barbara. She's asking, do the cannabinoids work in both UC and Crohn's? And what about the ileal Crohn's?
So in this case, we started by testing everything in vitro, just by using neutrophils outside of the body. And we are hoping that they are gonna be more useful probably for ulcerative colitis, but the idea is to test both diseases.
Thank you. Also, I wanted to ask, so there is a genetic component. You mentioned 5 to 10% of the patients. Is the genetic mutation usually in genes related to immune function, or are there other genes that might be responsible too?
Yeah, there are basically genes involved in the immune functions, such as interleukin-10. There are mutations in this interleukin that can lead to that are associated with IBD. There are also mutations in autotransporters, like one that we studied that is called PGP, that is in charge actually of secreting these endocannabinoids. So that's why we are also interested in these molecules.
Interesting.
But most of the loci that are related to immunity are those that relate to the microbe sensing. So that's why we always believe that there is that interaction with the microbiota that is like a feedback. You know, you have a mutation, you also have an altered microbiota, and that's cross-feed. Because not everybody that is genetically susceptible goes and develop the disease. There has to be a second trigger.
Thank you. Thank you. That was supposed to be my next question. Thank you for answering that. We have a question from Karen. She's asking, since genetics and diet can cause IBD, I was wondering what your opinion is about a mother with IBD breastfeeding their babies. She has IBD, and she was wondering if she has a lot of flare-ups in the last couple of months, but the baby is not growing as much as he should. Do you think my IBD can have an effect on this? And she's also inquired with his PD and her GI doctor. She just wants to know an opinion if you would have on this.
Okay, I think this is a better question for Anna because she's actually studying something related to this. So
genetics and diet, I mean, I think cause IBD is a strong word. You know, it seems that those two combined are factors that can cause IBD, but it's not the only factors that cause IBD. So I just want to, first of all, Karen, just to let you know that. So, and that's how it's so difficult to study, and how it's so hard thus far, even with all the research, to have a cure for it. That's why it's still incurable, because the causes of the disease are very hard to detangle one of another. So that's one. In terms of breastfeeding, keep doing it. There is studies that have shown that actually have been on medication and being in remission is beneficial for the mom and beneficial also from the baby and from the outcomes, and it doesn't pass through the birth to the breast milk, any of the medications you are taking. The baby not growing is something that we haven't studied, and I don't think there are studies around this. We are currently, that's what Mario pointed at me, because we are looking into that. We have a big cohort of pregnant women with IBD that we're intervening with a diet, and Barbara Oldensky here is the brain behind the diet that we designed for these patients. And we can tell you more maybe if Olicia and Sina invite me in 2 years, and we will have more response to that. But it's safe. What is known that is safe, so keep doing it, and don't stop your medication even when you're pregnant, because being on remission is essential for pregnancy outcomes.
Thank you, Anna. This is great. It's sort of the interface that we're looking at, where people get to know what current research is going on as well. And yes, we are booking you 2 years down the line as well. So the next question is from Lindsay. She's asking, are there specific cannabinoids that are the most promising for the treatment of IBD?
So yeah, so far we are using anandamide, which is the one that I showed. But we are working in collaboration with another team in Italy, and they are synthesizing synthetic cannabinoids. And the advantage of these molecules is that they are more stable than the ones that our body produces. So it's easier. We can make sure that they are actually going to reach the intestine, and they are going to be used to modulate this inflammatory response. So I didn't mention the names because they are basically a lot of letters that don't make any sense. But yeah, anandamide is basically our main target.
Sure, thank you. That's interesting. Do you need any sort of delivery agent, or is this something that can be taken as a tablet or so on?
Yeah, in this case, we did it directly in the media for the in vitro experiments. And I need to say that these compounds are so sensitive that they cannot be exposed to light or oxygen. And for the experiments we have performed with animals, we are delivering by intraperitoneal injection, but now we're planning to do it by oral gavage. It's a bit more complicated because we need to do it every day, so we haven't really reached that part. But yeah, we haven't seen effects, but we need to improve it still.
Interesting. Biplab is asking, what was the concentration of the cannabinoid that has been used in the assay? And do you think this concentration is toxic for the cells? And did you look at cell viability using MTT or so on?
Yeah, so I use it. The maximum concentration that I used was 100 micromolar, and that's one of the things that I need to do to test that it's not toxic. And no, it's not toxic at that concentration.
Sort of on a related note, how long do you think the effect would last? Is this something that you would need to keep taking?
Yeah, I think it's something that needs to be, as any other medication, needs to be taken continuously in order to prevent the activation of the immune system. I mean, to continue with this inflammatory response. But we still don't know. Those are things that we still need to check.
Interesting. No, it's definitely a very interesting approach.
I have one final question, perhaps. So how did you figure out that cannabinoids have an effect? Was that known before you started your project, or did you figure that out?
So there is some research, previous research in this regard, showing that neutrophils the neutrophils migration gets inhibited by these endocannabinoids. But there are so many other anecdotes that took us to this step. Like, we were usually working with neutrophils, and when we were drawing blood from patients, we realized that some of these neutrophils were not moving. And then by digging in, we realized that the neutrophils that were not moving came from people that were actually smoking marijuana the day before or the same day. So that was like a really interesting observation, and that's also why we think this is very promising. But it's important to highlight that also we want to modulate neutrophil migration, but we don't want to block it completely, because it's important to have this immune response is ready in case there is an infection.
Right, you do need them because they're there for a reason, right? So
Yeah, exactly.
okay, well, that's great. Well, if anyone else has any more questions, please type them in the chat box, and we can ask them later on. But for now, we'll move on to our next speaker. So our next speaker is Dr. Ana Maldonado-Contreras. Ana received her master's degree in microbiology at the Instituto Venezolano de Investigaciones Científicas and completed her PhD at the University of Puerto Rico. And I haven't shared my screen. But I will do that right now. Okay. So she's currently an assistant professor at the University of Massachusetts Chan Medical School. And over the last 15 years, Dr. Maldonado-Contreras' research has focused on pathogens and commensal bacteria residing in the gastrointestinal epithelium. Her current work focuses on designing diets and probiotics that can rebalance the gut microbiota and improve their health in IBD patients. Ana, thank you so much for joining us, and the floor is yours.
Okay, let me share. Can you see it? Thank you so much for the invitation, Alicia and Sina. It's a pleasure to be here, but followed by Mario that already did all the work, and I just have to tell you about diet. So as he said, like we believe, and there is no evidence more than epidemiological evidence that the increase of IBD or diseases such as IBD is due to industrialization, and part of them is the food that we consume. And if you look around, there is like this graph of incidence and how it peak in the United States, and 10 years later it peak in India or in China, for example, when there is this 10 years lag of adopting this westernized diet and westernized lifestyle. And it's again, like I said, it's all hypothesis and epidemiological suggestions, but it hasn't been proven because, it's, as I say before, the causes of this disease is very hard to define or comes from different flavors. So we in the lab focus in using diet as a treatment for IBD. And I'm going to share with you some of the latest research on diet for use it as a treatment for IBD. So, and I have to say, and if there are patients in the audience, I think the patients knew this before gastroenterologists and researchers thought about it, but you guys are the ones that have experimented with food and understand that if you eat something all the time, that food give you some symptoms. And in most of the patients that we talk to, when they talk to the gastroenterologist, the gastroenterologist doesn't think that diet have anything to do with it. And it has been in the recent decade or so that there has been an increased push to understand how diet contribute to the disease and can be used to manage symptoms. So, and as a disclaimer, like Mario did at the end, I have to say, food, we believe that food is an adjuvant, so it's not to be taken by itself, but it should be a treatment that goes with your normal treatment prescribed by your gastroenterologist. So don't drop the treatment and go on a diet, but definitely using a diet help. So let's see how this works. Okay, so the first study that I'm going to show you was done in Israel. In 2021 was published, and this was done in pediatric patients. There is a lot of evidence that enteral nutrition, which means a exclusive liquid formula diet, can help patients they're in a really flare up, and it's mostly children. They can achieve remission, as Mario say, which means they feel better, their inflammation is down, and then they can continue with their life. All the symptoms and the episodes just it gets regulated. Again, this disease can be cured, but it can be regulated, or the patients achieve remission. So this group in Israel decided to, but diet, the liquid diet is hard because it doesn't have too much nutrients, and it's not as appealing. So this group of researchers are trying to see if doing a diet that have solid foods and is complemented with this partial nutrition. The first one, where here can I put my laser? Nope. Let me change the laser point so you guys can see. Okay, here. So these are the two diets that this group were comparing in pediatric patients. This partial, instead of exclusive liquid enteral nutrition, just partial nutrition, and the other part of the nutrition was supplemented with a diet structure that they call the Crohn's disease exclusion diet, and it is characterized by having no animal or saturated fat or milk fat or gluten, emulsifier, red and processed meat, but include mostly fruits and vegetables. So half of the diet was this solid diet, and the other half was liquid. And then they compared to kids that were in an exclusive dietary enteral nutrition, which is liquid formula. They didn't find any difference in using one versus the other, but actually the one that it was whole food plus the liquid was more accepted for the kids. And they saw that only at 3 weeks of intervention, and after 6 weeks, most of the children improved symptoms, achieved the disease remission, and lower inflammation. And here I'm showing you the percentage of patients, and the number, I think it was 99 in each of the groups. So it is a sizable study. 83 to 85% of the patient at week 3 in gray or at week 6 in black achieve remission. So no symptoms whatsoever. decrease the symptoms, and 63 and 67 achieve remission, which means no symptom whatsoever. And then they found that up to 48% can have lower inflammation, and lower inflammation measured by one of the markers that Mario mentioned. There was like the number of neutrophils in the gut. So this is in pediatric children. What about adults? There is in 2021 also a group here in University of Pennsylvania tested two diets that are popular between patients with IBD, but there is no research showing that is effective. But these two are very popular among patients. Again, patients knew before we did. So they compared the Mediterranean diet, or MD, which include all fruits and vegetables, is low in consumption of red meat, and instead encourage the eating of fish, olive oil, not added sugar, and no processed food. And they compare it to the specific carbohydrate diet, which also include fruits, certain vegetables, is more restrictive in vegetables, and mostly excluding vegetables that have high starch content, like potatoes, for example. Only, not all red meat, but only lean meats, all fat oils, milk and grains are completely avoided or not included in the diet. And also there is a limitation in processed foods. So they saw, again, very similar output, or patients feel good with either diet. Achieve remission up to 43 and 46% of the patients that included in the study with either the Mediterranean diet or the specific carbohydrate diet. And this is at week 6. And then they found that they have a normal inflammatory marker in 30% of the patient, in 40% of the patient in either diet. So using one or the other didn't make much of a difference, but you can see this is a huge number for IBD. For achieving remission in IBD. The current treatment, first of all, is an infusion, and it is up to 42 weeks that you have to get infusion, and there's still the numbers don't reach this high percentage of achieving remission. And this is only 6 weeks on a diet, and almost half of the patient achieve remission. So it's very fantastic numbers in the world of IBD. Okay, how did I pass this now? Here, sorry. Okay, and then even after 12 weeks, participants sustain remission, and again, this one more close to 40% are still shows 0 symptoms when using the diet as an adjuvant treatment for the IBD. The other study, this is the 2 previous studies with patients with Crohn's disease. There is only this study with ulcerative colitis, and here only 17 patients were recruited for this study, and they compare the improved standard American diet, which are the basic guidelines for Americans to eat healthy, that is put up for the government, which means that people should consume 30 to 40% calories from fat, but then eat vegetables, fruits, and fiber. And then they use as a control or comparative group people that went into a low fiber diet, so which means most vegetables and fruits and only 10% calories from fat. So they didn't have a clinical trial. They compared the patients before and after the diet, and they only look at a dietary improve life, quality of life. So here is most of the patients actually that they recruited were in remission. So there is a little bit this partial Mayo is a survey to look at self-reported symptoms. So patients in the standard American diet or the low-fat diet have a lower percentage index, but it wasn't particularly significant. But because most of this seems to be a pilot study, so it was only in patients on remission, so they didn't have like active disease. But what they did was they have another questionnaire, is called the short IBD questionnaire, that measure quality of life, and they saw a huge jump in the quality of life of patients compared to the baseline when they ate the standard American diet or the low-fat diet. Then this included, they have more physical mobility. They were better emotionally in solving problems. They seem to have better social function and less body pain. So those are measurements again of diet quality of life. Sorry, and the other thing that they measure was the microbiota. They want to see if this diet changes the gut bacteria. That is, as Mario says, one of the components of the many factors that can promote onset of IBD. And what they found is that the going into these diets increases alpha diversity, or like the diversity of the gut. So there's more different bacteria, and that is good. Usually a high diverse microbiota in the gut means that a microbiota is strong and healthy. So it's not significant, but we see a trend of going up after the diet. And here then they found a specific bacteria, then they went narrow it down to see which are the bacteria that are changing before and after diet. And they saw a decrease of actinobacteria, which again, we call it dysbiosis, but it's an alteration of the microbiota with this patient have. And the IBD patients UC patients usually have an increase of this bacterium that is related to a more pro-inflammatory signaling. And we see it that after either of the diet, we see a decrease on the number of these bacteria in the gut after the diet, an increase of Bacteroidetes, which are health promoter bacteria, same as Faecalibacterium praunici in Prevotella. Again, both bacteria that has been seen reduced in patients with ulcerative colitis, and after a short dietary intervention, we see this bacteria going back to normal levels when healthy, not normal, when other healthy people have values. And then in our lab, we partner with Barbara Lansky, that has created a diet for IBD based mostly in the carbohydrate restricted diet and in the Mediterranean diet. She mixed them up, and with her knowledge in IBD and listening to the patients, if somebody have taught me how to do that, is her, created a diet focused on changing the microbiota. So the diet, we call it IBDA, or anti-inflammatory a diet for IBD, and it's based on 4 components. Inclusion of prebiotics food, which means more fiber that increase the good bacteria in the gut and sustain their abundance in the gut. Probiotics food, that is more of fermented food that have good bacteria in itself, like yogurt, kefir, in these very popular products lately that have been very popular lately. Beneficial foods, because most of the diets that I shown you before are very exclusionary. So there is a lot of exclusion of tons of food, so they can only be eaten in short term, because then after that you have to change the diet to get a proper nutrition. So we are also including an arm of including food that are beneficial for the individual. And then we do exclusion of foods, and we like to call it that instead of exclusion, we're substituting foods. So the foods that are excluded are the ones that are known to cause GI symptoms. And what I believe is very relevant is that we really try with Barbara's team to find substitutes for these foods. We're not just telling the patient, don't eat this, this, and this, but instead of this, you should try this, and we will teach you how to cook it. So I believe, of course, I'm the one doing the research, but I believe our research is very cool because we also take into account the patient and their needs. You know, it's very hard to tell a person what to eat without giving them instructions and without giving them substitutes, because it's very attached of what we do every day. So it's hard for us to change overnight the way we think about food. So we actually, besides telling the patients what to eat, we teach them how to cook. And here are some pictures of us and our nutrition team. And at some point, we have also a chef coming to our teaching kitchen here at UMass Medical School and teaching the patients how to cook the recipes that we're telling them to cook. And, you know, you're not using sucrose, you're not using sugar, but then how you sweeten your food, etc. So we give them the spin, and then we actually give them hands-on experience so they can adopt this diet, not just for 10 weeks, for 6 weeks, but it's a change on their lifestyle. And I think here they're pictures some of the patients, and also Barbara has I don't know how many years ago, but she's also started teaching medical students on diet and in nutrition, because it's very important that they recognize they don't have the skills, they don't know that is not being taught in medical school. So she is a pioneer changing that in our school and teach the medical students also the importance of food and actually, you know, cook instead of like just ordering pizza. And also we are trying to get trying to keep with the flows. So we created a website, and I'm just showing you here, but we also have Instagram pages, Facebook pages, so the patients have a readily accessible recipes and tips for cooking. We partner for this study that I'm going to show you. We partner with, as I say, with a chef, and the chef did a video going to the supermarket, you know, and like teaching patients how to find the food that they're looking at, you know, how to read labels, etc. Because it's very confusing. Going to the supermarket is just so many options, and so what to choose. So we'll help the patients. We whole handing them to really, you know, it's not an easy task. So we're trying to make it simple as much as we can. Instead of giving, we say that instead of giving the fish, we're teaching them how to fish. And with those instruments. And we have a pilot study that we did with 19 patients. This is just some of the studies that we have done here. I'm showing you actually first. Barbara in 2014 showed that using this diet reduces the patient's symptoms, and this is for Crohn's disease. Again, this an activity survey that we give the patients to fill up, so they tell us about their symptoms. Then there was a completely reduction of the symptoms for patients with CD, and this is only 2 patients, but in those 2 patients with UC, we also saw a reduction of the disease activity. Later on, we did a study where we recruited 19 patients, and we collected samples before and after the diet to look at inflammatory markers. We also assess self-reported symptoms before and after the diet. And here I'm showing you down here the self-reported symptom. 99 out of the 18 individuals in this case reported a good improvement of their symptoms. And it goes along the lines of the other study with 40 to 50% of patients that go into a diet experience a reduction in the symptoms. 7 didn't have any, they didn't feel better or worse going through the diet, and 3 have adverse events, and 2 of them was not related to the diet. They have infections and other causes that wasn't related to the diet. We also see a reduction in pro-inflammatory cytokine like IL-28 before and after the diet. We see a small reduction of these. This is only 8 weeks on the diet. And then this other cytokine has been actually associated with improvement of IBD symptoms, and we're seeing an increase on that in the patient that we collected blood for. And here we have another measurement of inflammation, and this is fecal calprotectin, which measure the neutrophils that Mario was talking in the gut. If we can measure it on the stools, and we see that it's very high in all the patients at baseline. And after, again, 8 weeks on the diet, we see a dramatic reduction of the levels of inflammation, of fecal calprotectin, which refers to inflammation in the gut. So we believe that this diet is also working. We also look at the microbiota, which was one of our main interests. Because Barbara claimed that they changed the microbiota. So we're like, well, let's look at it. Let's look if that's the case, and it actually is the case. So I'm showing you here just one patient as an example. So each dot means a stool sample. In red, the samples that were collected at baseline. And if they're grouped together, that means that they look more similar to each other. So they have the same bacteria. And if they're different, they're very far away. That means that they're different than the other group. And here we see in red how the microbiota looks like. In that, it was grouped in that corner, and as soon as we start the diet, the microbiota start changing, which are those blue dots, changing direction and then getting together in one cluster. And same with the ulcerative colitis. These are again in baseline. All the stool samples look similar to each other, but different than the samples that were collected at the intervention. And then we look at which are the bacteria that are driving these changes in being similar or dissimilar. Sorry. And most of the bacteria are related to Bacteroidetes, as I told you before, is a health-promoting bacteria, and it's reduced in patients with IBD. So we see these samples on baseline and samples on intervention. We have 19 patients. We collected over 500 stool samples throughout 8 weeks of intervention, and we see an increase of these Bacteroidetes vulgatus, Dore, and then some Clostridia bacteria that also is very, they're very few in IBD, and they're associated with severity of the disease. And after, again, 8 weeks of intervention, we can switch the microbiota and increase these health-associated bacteria. So how much time do I have? Because I keep talking.
We usually end around at 7:30, and we want to have some time for Q&A.
Okay.
So.
Okay. So so what all these diets has in common? Because, I don't think my diet is better than any other diet. I think the way that we apply perhaps is better and more patient-oriented. But what do they have in common? They have in common an increased consumption of fruits and vegetables. There is a reduction or complete elimination of processed food, reduction of elimination of sugar intake, reduction of animal protein, increase of fiber consumption. And here I will have to put, these have to be with a dietary guidance, because patients with IBD need help in digesting fiber, and low fat, and modifying these fatty acids that they're more monosaturated fatty acids in our diet. So we believe diet with these pillars works as an adjuvant treatment for IBD. And this is the people in the lab and our collaborators and our team, and where the money comes from. So happy to get some questions.
Great. Yeah, thank you so much, Anna. That was a really nice talk. So maybe I'll start with a question. So were the patients that received the IBD-A diet, were they on medication for their IBD when they started the diet, and what was their IBD well controlled, or no?
So in the pilot study that should I stop? Okay, now. In the pilot study that we did, we have some patients in remission, and we have some patients with a moderate disease, but they were all in their drugs, and we control for all that when we're doing our analysis. Mm-hmm. So the only differences between before and after was the addition of the diet, no other medication.
Right. Okay.
Thank you, Anna. That was very impressive. And it's amazing how much work you and Barbara and the entire team are doing to actually help patients to even follow the diet, because that is definitely one of the most challenging things as well.
Very much.
I'm just thinking about myself going to the market and I'm like, okay, I want to get this and this and this. And yeah,
And
this
you go exactly to the same aisles. You cook, if you think about it, you cook more or less the same meal. So just changing that mental structure is what we're trying to achieve.
this is a very important work. I just had a small question. So what role do probiotics play? So is that something which works as a good adjuvant therapy?
In pills, you mean?
Yeah.
So we haven't tested that, and there's other groups that have tested that and have found no very promising results or mixed results. So that's why we don't include them. But we are believers of the probiotics foods, the fermented foods that and there is epidemiological research that supports that. And that's what we believe that there's, and there is also actually a study from Stanford that shows how probiotic fermented foods help to maintain a balanced microbiota and then modulates the immune tone on the patients. And these were healthy control, actually.
Interesting. I was also thinking of Mario's presentation where he had the images of the colon, which looked like completely disrupted in IBD. So I was wondering, is there a stage at which the diet started would obviously be more effective because the tissue regeneration might not be as efficient? Later.
So there is this diet, for example, have several phases depending of the disease severity and the symptoms that you're presenting at the moment. The diet is customized to your needs. And that's what I like. The fiber, for example, don't go and then, when you're in a flare up and start eating fruits and vegetables, it has to be cooked. It has to be blended. So there are phases on the diet. And as you progress, you can change the texture of the foods and the way of cooking it.
Interesting.
And that's also one of the things that some of the previous the big clinical trial has failed to do is like, okay, they're in remission, now what?
Personalized it.
You send the patient to the house, and now what? What do they eat? How do they eat it?
Mm-hmm.
So I think we're trying to work on in this phase stage so they can achieve until phase 4 when they can eat mostly everything on normal, more like whole foods without so much preparation.
Interesting. I would imagine you've got an app coming out as well at some point to help patients track.
I'm sorry, say that again.
An app to track the diet and.
Oh yeah, that's
yeah,
exactly. And another exciting thing that I think we're doing, this is like a blueprint for other diets, because diet is very culturally attached. So this diet is being proven here in our Caucasian white patients that have access to the foods that we have in New England, right? Or Barbara actually tried to make it nationwide. But then what are other patients in other regions that eat different and have different food affordabilities or even access to food. So we're now working with a team in Puerto Rico, and we're using the same basis of this study on how which food modulate which bacteria and which food make a patient feel better. How can we do, instead of peanut butter, avocado toast, or change it what is available and what is palatable for the patient, so there's more compliance.
Sure, yeah. And this is not something easy to test on model systems, right? You have to, yeah.
And so we go from humans to patient. Then now we have, I don't know, Maya, how much? 4,000 samples. Oh, and wow. I also, my postdoc that joined 3 weeks ago, have tons of things to do and test in mice.
Yes, at least 4,000.
Excellent. Well, Thank you so much, Anna. That was excellent. And another big thank you to you, Mario, for your talk. And thank you, everyone, for tuning in and for asking the questions and participating. So we will post the event survey in the chat. So if you have a minute, please take that. We will also send that in a follow-up email. And again, please tune in into our upcoming talks. We will follow up with some emails about those. So thank you so much to everyone, and we hope you have a good night and enjoy the beautiful spring weather.
Thank you. Have
Bye-bye.
a good night.
You
Thank
learned a
you.
lot. Thank you so much.